So this is Miss McKenzie. I want you guys all to meet. She's going to be here with us for the rest of the semester and definitely into April. Maybe May. We're not really sure what MSU has planned, but I'm going to let her take a couple minutes and just tell you a little bit about herself and how she ended up here with us.
Cool. Well, hi everyone. I'm Taylor. You can call me Taylor or Miss McKenzie, whatever feels more comfortable for you. Well, I'm a senior at MSU, and I study biology, and I would like to teach high school. I think biology, but also chemistry and other kinds of science as well. I am here, I guess, because I am going to be watching Miss Snook and working with all of you guys so that I can practice my own teaching and think about what I would like to do when I have my own classroom someday. So I'm excited to learn with and from you guys, and I'm looking forward to the semester.
Yay. You guys want to say hi? Welcome? Give her a voice.
Welcome to the most fun class ever.
Nice. I think that we are so lucky to have Miss McKenzie with us because there will be like a second person that you guys can ask questions or clarify. I think during breakouts, it's going to be super helpful to have a second individual in there, as well as during that flex time and you guys want to come in and get some help. And the other thing I thought was beneficial is that Miss McKenzie has been learning the same as you via the internet. So I thought that you might be able to relate to her and she can relate to you guys with what you're going through, you know, the difficulties of learning online and being so much more independent than you have had to be in the past. So we know especially in AP, we're already going to be a lot of independent learning, but this year especially, we're doing so much more. So I don't want to take up a ton of your time. Okay, so you guys, I wanted to do a little bit informal at the beginning here, so I don't have a slide up. Have you done any sort of chromatography before? Voices are good because you got the floor. Anybody do? No? Anybody have AP chemistry last year?
Oh, I'm having it right now. So we just did the lab yesterday.
The chromatography lab?
Yeah, on Monday we did it 2 days ago.
Shut up.
Yeah.
How? So that was lab 5 for you.
Yeah.
And lab 4 for us. How crazy is that? It's in both similar. We're working specifically with plant pigments, but the whole concept of chromatography. I linked the AP Chem 5 video to this because if you really wanted to understand chromatography and how it works, I thought that was a good resource. So I'm going to give you some shortcuts. You know, we always visit the lab bench and we do, like you have that tutorial and it takes you step by step through the lab you're supposed to do. So this is something everybody should do. It gives you a little practice quiz at the end. But since we're not doing the lab and I'm going to give you kind of a shortened version of it, you should still go to the lab bench and go through the step-by-steps. Okay. Of course, Bozeman, we always go to Bozeman to watch lab walkthroughs. So he gives you a rundown of that as well. This worksheet here, this is going to be your dry lab, the 2018. I actually made it online for students who were absent or needed to make up a lab that they weren't there for. So I made it a couple years ago, I made it available online. So that's the one you're going to fill out. And I am just going to talk through our agenda slide. I just put it all on the agenda rather than making a separate PowerPoint. So I was thinking you probably have done paper chromatography at some point and you just don't remember. Maybe you took a coffee filter, drew on it with black marker maybe and put it in a cup of water. Does that sound like anything you might have done any time in your life? We used to do it in Impression 5. We'd make butterflies out of it. I would do it sometimes with my biology class when we study water. That would have been a good one for us to do when we studied water for a day. Okay, so I'm going to minimize you guys a little bit and put you over here. Now, if you want, you guys are free to not show your face because I'm only going to see 5 of you at a time anyways. So we're looking at pigments, plant pigments in this lab. And there's lots of different pigments, which we're really familiar with right now because of the changing leaf colors. I found this graphic this morning, and I just really liked how it was a flow chart of pigments that are related to photosynthesis and then pigments that serve other purposes. And there's more that you could put on there, like flavonoids. But we're familiar right now with chlorophyll A because we've been talking about the light-dependent reactions, and that photosystem 1 and 2 both use chlorophyll, right? So there's 2 different versions, chlorophyll A, chlorophyll B. They're going to absorb different wavelengths of light. Right now, we're seeing those red, orange colors, xanthophylls, carotenoids. So carotenoids come in 2 versions. So you kind of refer to the xanthophyll as the yellow, red, orange, carotene, more of the orange. So we see these in the yellows, but we also see them in different parts of plants, like the fruit, you know, they come in different colors, like the yellow of a pepper or the orange of a carrot. And the fact that plants have multiple pigments makes it so they can be more efficient in photosynthesis, right? If you can capture different wavelengths of light, sorry, my cat needs in. If you can capture different wavelengths of light, then you're going to be using more of the spectrum, more of the white light that's coming to you from the sun. And so you're able to capture more energy. So that is one of the benefits of having multiple pigments. So you've referred to like chlorophyll as the main photosynthetic pigments, and then the others are accessory pigments. Okay, so that's just some background. So we're gonna do 2 different pieces to this lab. Chromatography separates pigments, in this case, other molecules if you're in a chemistry lab. And we're gonna be looking at some specific pigments, chlorophyll A, B, carotenoids, and xanthophylls. And you can see the pigments are different sizes, right? Different molecular shapes, different molecular sizes. They're going to have different properties, and that is what allows us to separate them out. And we're using a form of paper chromatography to do that. We're taking advantage in our second lab of the absorbance spectrum, their ability to absorb different wavelengths of light. And so again, you can see chlorophyll A, chlorophyll B absorbing. This is the absorption, right? So this is what they're taking in. We know that we see pigment colors by what's reflected. So we can see they're taking in colors down here in the blue-purple area, which means they're reflecting the rest of the colors, which is why we see green from the chlorophyll, because we're seeing the color that's being reflected, okay? So we want to be able to read that type of a graph. In this particular, the first part of the lab, you're doing your chromatography. This, we would use spinach leaves if we were in class. This is not a spinach leaf, but it's a good graphic, I think. So you would put it into the little cup, I forget what it's called, and you would pulverize it. Or another way to do it is just roll a quarter back and forth over top of it, on top of this chromatography paper. So you would have this chromatography paper and you would put it in a cylinder and you'd use some kind of organic solvent like acetone. So you can try this at home. Maybe try it with isopropyl alcohol, because you probably don't have, you might have acetone at home, but anyways, you could try it with a filter, like coffee filter paper. So you're gonna put a dot of your spinach solution onto the paper, okay, above the solvent. And then as the solvent is attracted to the water, right, adhesion, capillary action causes that solvent to move up the paper. The different pigments will dissolve in the solvent. Okay, so then the solvent acts to carry those pigments up through the paper. Then they have different affinities for the paper itself, different attraction strength to the paper itself, and they will settle out at different points, okay? So this allows you to separate pigments like this through chromatography. So this is the basic setup. You have your solvent at the bottom. It's going to be climbing the paper. Where it, like you would give it a certain amount of time. So if you ran it for 20 minutes, the height that the solvent traveled would be called the solvent front. All right. And then the pigments, as you can see, will settle out at different positions during that 20 minutes. So you're going to run it for a certain amount of time. This is really wordy. It's just explaining how it works. The filter paper is a stationary phase. The acetone is a mobile phase. So the acetone dissolves the pigments and carries them. So that's the mobile phase. And then depending on how strongly they are attracted to the solvents and how strongly they are attracted to the filter paper, they will settle out or stop moving at different positions. And this might be due to their affinities, it might be due to their molecular weights, and then that difference allows us to separate pigments or other chemicals. That's the first lab. The second lab, you're using spectrophotometry, which is kind of fun. You're looking at two different things, absorption and transmittance. So absorption is how much of light is going to be absorbed by the solution. Transmittance is referring to how much passes through, okay? So in this case, we have a light source, and you can select different wavelengths of light when you use the machine to pass through. You're going to have a spinach solution here in your cuvette, and the light is going to flow through to a detector. So depending on what wavelengths are able to pass through, you know the other ones were absorbed, okay? So then you're gonna make an absorbent spectrum, which is what you saw before. So this is what the spectrophotometer would look like. We select different wavelengths of light here. This cap opens up, and we would put our cuvette in there, okay? And then when we shine the light through, certain wavelengths of light will get absorbed, and others will pass through and land on a detector. This is why it's relevant to photosynthesis. So remember in the light reactions, we had our 2 electron transport chains. The one went to make ATP, the other one went to make NADPH. You labeled on a worksheet last I saw you guys, Monday, you labeled NADP reductase. Remember, NADP plus reductase. So in photosynthesis, you use NADP, or you don't physically use it, right? But plants use NADP to be reduced. And so that energy of the electrons moving through that electron transport chain is going to reduce NADP into NADPH. In the lab, you use a different chemical, DPIP, to replace NADP+. Okay, so DPIP in its normal color, it will be blue, right? So imagine blue is going to absorb more wavelengths than clear. So there'll be less transmittance if DPIP is blue. Electrons, when stimulated, we know they get excited, they jump out of their chlorophyll pigments, and then they travel through and they reduce NADP. So if you replace that with DPIP, when the electrons are excited, they end up reducing DPIP. So DPIP will turn clear when it's reduced. So this is just an indicator of photosynthesis occurring, right? This is the amount of energy that has been absorbed. So if DPIP changes color, we know photosynthesis is occurring. Basically, that's what's happening. And then we're going to measure how much light is transmits through the fluid, because the more that it can go through, the clearer the solution is, meaning the more photosynthesis has occurred. Okay, more energy has been absorbed by the pigments of the chloroplast. So these are your setups. All of them will have, well, 3 of them will have DPIP, and then 1 will not. Okay, so that's going to be your control. This shows you what it would look like if there was no reduction. You're going to have unboiled chloroplasts. So this is a positive control. It shows you what things should look like if photosynthesis is occurring. Here's some experimental setups. We have, this one is wrapped in foil, so it's not going to absorb any sunlight. So there shouldn't be any photosynthesis occurring. DPIP boiled chloroplast we know that heat should denature the chlorophyll, right? So the chlorophyll should not be in a state that it would be able to undergo photosynthesis. Okay. And then the last slide, this is an FRQ based on this lab. So the AP Bio test often grabs the labs that you do. You'll have at least 1 question about your lab. And you should know why each piece was included. Why did you use DPIP? Why did you use chloroplasts that were boiled? Why did you wrap it in tinfoil? You should be able to talk about standards, what was held constant between all of them. You should be able to talk about positive and negative controls. What was your independent variable? What did you measure? So you were measuring the transmittance, right? What was your independent variable? What was the thing that you changed? So you might have changed the amount of sunlight, or you changed the condition of the chloroplast. That's another day. Okay, on this PowerPoint, I also included some, I also included like links where I got pictures, but there's videos. So like this video explains the lab. There was a couple different videos that I thought helped with that particular concept. So if you wanted to, you can always look in the speaker notes down below, okay? So that's the basic concept of the 2 parts of the lab. All right, so you guys are gonna pull this up, and directions are included, but people usually have trouble just wrapping their head around that DPIP especially. You're gonna have data that has already been collected, so you're just interpreting, okay? So in this case, we need to calculate the RF value, which just basically refers to the tendency of your pigment to move compared to the solvent itself. Okay, so that's gonna be characteristic of certain pigments. So here you're just calculating that RF factor. Take that same RF factor, now determine which pigment that RF factor is for, okay? So like this doesn't use the name of the pigments, it just gives you the color. So like blue-green, you're gonna calculate an RF value for, and now you'll know what the RF value is for chlorophyll A, because that's your blue-green color. Okay, so now you're just matching them. This is not in the same order as this slide. So look at the colors, match the colors. Xanthophyll, yellow. Yellow, green, chlorophyll B, okay? Yeah. And then you're just interpreting those results. And then this is getting to that spectro lab, okay? You got your results. All ran for time, so that was an independent factor there. And then you will be graphing that one. So again, remember I double click and I'm able to graph on the graph that I gave you. Be sure to label X and Y's, and not just like what is on the X and Y, but your increments, okay? You're gonna have 4 different lines because you have 4 different setups. So use different colors maybe. You can, those of you who have mastered the whole concept of using the Google Sheets and cut and paste, you can make a graph on Google Sheets using this data just plug in the data, create your own graph, and then copy paste it in place of this one. And then just interpret the results and answer the questions. Okay. So what questions do you have?